I'm going to briefly cover some of the topics that we're going to be discussing for this program on genomic data analysis. So the objective of this program is really to cover a broad overview of different types of topics, provide you with the methods that you can practically apply to analyze the data, genomic sequencing data, but also importantly to understand how this can be applied to a variety of different project examples and try it out on your own. So hopefully by the time that you complete this program, you will understand what the terminology means, especially when you read the papers. You will also be able to practically have experience and know where to start your project, how the different stages are connected to each other, how to interpret this, and have a meaningful discussion with your peers or with others about genomic data analysis and interpretation. But today I want to briefly start from the very beginning. Later on, I will show you where you can find a lot of the information that we cover in today's session. But I will just take a few minutes to talk about some of the basics, because we typically assume that everybody's on the same page, but a lot of people, just by not understanding certain types of terms or maybe concepts, it's very difficult to follow along. So what is DNA code? What do we know about the DNA code? And what is it that we still don't know? What is there else that we are trying to learn? Why do people continue to study genomic data and try to interpret and understand what this code means? So DNA code, especially when we talk about eukaryotes and we talk about people, we think of this code as a book of recipes. So it's organized in a specific structure. It has sentences, paragraphs. Paragraphs are organized into chapters. And so really learning this book and how to navigate and spot these patterns is the basic approach of genomics. So DNA is short for deoxyribonucleic acid. It's a long molecule that's composed of two chains that coil around each other and form this double helix that we all know. And the code contains instructions. So these instructions are coded in the human genome in about 3 billion base pairs. So it's very long. Not all of those base pairs contain information that ends up being translated into proteins. And similar approaches of putting information into this code are used by bacteria, viruses, and large organisms, multicellular organisms as well. So the same approach, the same code contains directions or recipes for what we call life. And life is the condition that, that distinguishes animals and plants from inorganic matter. So what does life give us? It gives us the capacity for growth, reproduction, functional activity, and continual change that precedes death. So when we talk about life here, you can see a really nice animation, or actually not an animation, a video, where a single, small microorganism, and maybe even different kinds of microorganisms, are moving, right? And that's really what distinguishes life from these inorganic matter, like rocks, for example. So when we have these, these recipes or these pieces of code, they are organized in a very tight way by compressing this long molecule into a very tightly compacted kind of nucleus. And when cells divide, what happens is that in the nucleus, we have all of this DNA, and when it divides, we can see how this DNA is organized because it needs to be unfolded. And so what we discover are these strings that are inside, and we can actually see them. So this is the process of division, and what you can see are these chromosomes, right? So these chromosomes are actually, string of DNA, also packaged very tightly in these specific shapes. So these chromosomes contain different genes. Each one of our cells contains 23 pairs of chromosomes. One of the chromosomes comes from our mother and the other from our father. And the unique structure of chromosomes keeps DNA tightly wrapped around spool-like proteins called histones. Without such packaging, DNA molecules would be too long to fit inside our cells. So when we think of this chromosome we can expand it, and this is what it looks like. So here's the actual DNA, this string right here that is kind of coiled around these histones. So if we continue and unfold it, you can see that these are the histones, and it's packaged right around those histones. So when we speak about this code, and what does this code actually contain? What instructions does it contain? So the product of the code and the way that it interacts with the environment is called a phenotype. So a phenotype can describe a single cell, a tissue, organ, or the whole organism. Phenotype of an organism might include height, size, and activity, and phenotype of a cell might include its specific function, size, and morphology, or its shape. So the code, which is packaged in this molecule, defines this phenotype, right? Defines first phenotype of the cell, then of the organ or the tissue, and then of the whole organism, right? And so the basis of this code is all about these nucleotides. So these nucleotides are made up of a sugar called deoxyribose, a nitrogen-containing base, and a phosphate group. The nucleotides are joined to one another in a chain by covalent bonds that form between the sugar of one nucleotide and the phosphate of the next. And the result of this structure is an alternating sugar-phosphate backbone. So each strand of DNA molecule consists of a sequence of bases that is complementary to the bases in opposing strand. So when we say base pair, right, you will hear a lot when we speak about genomics, this term called base pair, nucleotide, amino acid. This is where it comes from. So a single structure here is a base, and the connected are base pair. This molecule can be read from both sides. So you can see the 3' and the 5' ends. So you can read from either side. And this single base pair represents some kind of a nucleotide. So this nucleotide, we would refer to one of these adenine, guanine, thymine, and cytosine. So to understand how this code works and what is in this code, as you all know, this is the famous starting point of the Human Genome Project. You can see it was published in the New York Times on Tuesday, June 27, 2000, when they announced that this code of human life is cracked by scientists, right? So now scientists understand how these DNA sequences that we can now read are organized to inform us about life. What is life? How does life function? How does it produce the phenotype? Now, today, we are in 2021, 21 years later. So if that happened 21 years ago, what else do we need to learn about this? So it turns out that simply reading the code and annotating it is a much longer process than the human project itself. So the human project itself was focused on understanding what is in the code. And they discovered that there's only 1 to 2% of protein coding genes. The rest are non-protein coding genes. And so we have a lot of other types of information that is in the DNA. And we now know that there are a lot of questions, there are a lot of additional things to understand about the human genome, about the DNA, how it's organized, and how it actually works. And it's a very complex approach that people now take to study individual specific regions of the DNA and try to understand what is the impact of how this code is written to describe life and describe phenotype. So as we spoke, DNA is stored in the nucleus, organized in chromosomes, and specific regions are responsible for specific functions. Some of the easiest regions to read and understand their function are what we call protein coding genes. There's a total of 22,500 protein coding genes, but even that number continues to change. Protein coding genes are the sequences that are used to produce proteins, and they also have a specific structure. So when we think of this code working or producing that phenotype, the way it is done is by DNA being transcribed into RNA and then being translated into proteins. So these proteins that are produced from DNA is facilitated by several different proteins it's a complex kind of a translational process that we will study in a little detail to understand exactly how it works. But the basic concept that we need to understand is that the individual nucleotides or base pairs that we saw in the DNA eventually have to be converted into amino acids, which is what the proteins are made up of. And so when we think of protein coding genes, we're going to be looking at sequences, dividing them up into tricodons, and then looking at how those trinucleotide codons, and looking at what they mean, how do they function in terms of amino acids. So the function of the RNA polymerase is to convert DNA to RNA, and then the ribosome, of course, converts the RNA into protein. So the genome composition, let's just talk a little bit about how the human genome is maybe different from other genomes. The human genome is not the longest genome. You can see right here's the human. Grape, for example, is much longer, not much longer, but longer nevertheless. And influenza, for example, flu or COVID, we all know is the thousands of nucleotides long, as opposed to the human, which really has tens of thousands of genes and billions of nucleotides. So influenza, for example, has 11 genes, and is somewhere about 20,000 nucleotides, and that's how many genes we have. So here you can see what types of genes do we actually have, and you can see that 9%, so almost 10% of genes, we don't even know what they are. But how do we know that a gene is a gene? So a gene is also organized in a very specific way. They're not just sequences of nucleotides. They have upstream and downstream regulatory sequences that control transcription. Inside the gene sequence itself, there are codons and introns that undergo splicing before a gene is translated into a protein. And the length of these genes can vary significantly. And so they vary because they have these introns that could be very long. So when we speak about translation, like we said, the translation occurs as a result of ribosomes. So here you can see, this is a microscope image of unfolded DNA. You can see that the ribosomes are these large dots right here, and that is what they look like. So what happens is mRNA gets into the ribosome, and the outside of this ribosome, we already have this protein, which is a sequence that is folded and eventually forms a protein that looks like this. So this protein, this is actually two different proteins bound together because proteins now have a function. They interact with each other. And so in the context of genomic data analysis, we can look at every step of the way. We can look at the DNA. We can look at the impact of this DNA on transcription. And we can look at the impact of DNA in terms of translation. And so the ultimate goal of genomics is to integrate multiple aspects of this kind of data by looking at where the gene or genes are located in terms of the chromosome. What do we know about the organization of that gene and how that gene could be translated into a protein? So here you can see that protein structure. And eventually to validate its function through experiments. So here you can see this is the same protein. This protein is NM23H1. This is a protein that is responsible for control of metastasis or spreading in cancer. And you can see that its structure is known because we have a crystallized structure of the protein, but we also have studies that show how this protein functions, and we can measure this in terms of animal models, cell lines, and compare and contrast how it works in different variations. So when we talk about genomics in this program, we're going to be looking at data to try to understand what this data can tell us about a particular phenotype. And there are several different ways that we're going to be able to look at this. We're going to be looking at methods like multiple sequence alignment, variant calling, phylogenetic analysis. We'll talk about what types of mutations and variations we can find, which ones cause a change at gene expression level, which ones cause the change at the protein function level. So we'll talk about all of these things throughout this program. But the basis of this program is going to be based on data. And the data that we now study to represent genomic variation has evolved since the beginning of that human genome. And now we have thousands and thousands of human genomes, as well as genomes that are representative of pretty much a lot of organisms that you can study. So when you think of what you can do with genomic data analysis, once you know how to analyze it, you really have a huge potential of understanding validation of specific types of variation that we can observe, including human disease and response to different treatments. And ultimately, this trend will continue. So knowing and acquiring the skills to analyze such data is a critical skill that you will take away with you to be able to contribute to the continuous exploration and understanding of how genomes work. What do they encode in terms of genes, and how can those genes be studied in terms of regulation of these complex conditions like cancer, like infectious diseases, like neuroscience, and others. So after you heard this brief introduction into the topic of genomics, I would like to pass it on to Dr. Raghav, who will speak about the other sessions in the program and kind of talk about more of the practical